Hello everyone. Today in this lesson we'll be learning about meiosis. So yesterday in class you guys watched a video. You watched the funnier side of the video or of this topic. Think of this as like the nuts and bolts or the detailed version of that video. What I'll have you do is there's a packet, it should be on Sister's desk, that has two presentations, with the blanks. We will go through the two slides and try to keep this under 10 minutes. You also will get, I know some of y'all will hate me for this, but I'm giving a thick packet over break, and part of it's not because I want you to be miserable, it's just that I want y'all ready for the AP exam. So I'm giving you this thick packet. I don't know if we're going to get to go, I don't know if we're going to be able to finish all of it, but what I'll have you do is like once a day, at least to keep consistent with the schedule of the AP. I'll give you a 10-minute video every day so you can fill in the parts for that. I know it's a massive, it's a gigantic packet, but for some of you that are taking it, I want you to be very well prepared. So you'll be receiving that today also, or maybe some of you are getting it on Friday, but it's a review packet, and over break you'll be listening to a lecture every day. I promise you I won't go over 10 minutes, but I'm going to try to get through a lot of stuff in a short amount of time. So let's get to this video. It's on meiosis. Again, you watched the video on this. You should have learned this in regular bio, so I'm going to go through this relatively quickly because this will be kind of like the third time you're listening to this stuff, but again, I have to go over these details. So first of all, again, you're going over this in your notes and filling in the blanks. So genes, if you haven't heard of a gene before, it's a segment of DNA that codes for a basic unit of heredity. So remember, the code for DNA is like A, T, G, and C, so a specific sequence of A, T, G, and C it codes for something. That's a gene. Okay. Offspring, or the kids, acquire genes from parents by inheriting chromosomes. So this isn't my family, but consider this like a mom and a dad, and they give quote unquote their genes by through chromosomes, which we talked about earlier. There's two ways to do this. One's asexual and one's sexual. As you may guess, asexual means just one parent, and sexual just means two parents. Asexual, when you have offspring, you actually create clones, or your offspring is a duplicate of you. It's like having a twin. Obviously, it should be exactly like that, or exactly identical, because you're not having a male and a female like merge their genes together. Asexual, you only have one parent, so like you just pass on those genes. You pass on the same DNA to your kids. So asexual produces clones. Sexual produces gametes, or sex cells, meaning sperm and egg for humans. Asexual, single parent. Sexual, two parents. Asexual, little variation, meaning like there's not very many differences between parents and kids. For us, there's lots of variation and diversity. Some of you may have parents or cousins or brothers and sisters that look completely different than you. Asexual is fast and energy efficient. Sexual is slower and uses a lot of energy. Asexual, we're talking about budding, which I'll show you a picture of. Sexual, talking about humans, trees, stuff like that. So here's a picture of it. This is asexual. Here's the parents, and all of a sudden, when we talk about budding, I'm talking about like a piece of the parent just like sort of like branches off and then becomes its own thing. So that's what we call about budding. And sexual, meaning like a male and a female counterparts merge together to make an offspring. So a couple of basic definitions. Somatic, we're talking about a body cell. Okay, a somatic cell is for humans 2n, or 46 chromosomes. Okay, each pair of homologous chromosomes. Homologous, may know the prefix homo means same, and chromosomes, meaning where the genetic code is one chromosome from each parent. So I'm going to show you some pictures. Autosomes. So of these. So there are 23 pairs of chromosomes. So look at this, 2n equals 46. There's actually 23 pairs of chromosomes. 22 of them are called the autosomes. These autosomes are the chromosomes that cover everything. So they code for everything about you. So you may be wondering, hey, if there's 23 chromosomes and 22 of them are autosomes, what's that last pair? That last pair is the sex chromosomes, and some of you may have heard this before, but the females are XX and the males are XY. So the gametes, n equals 23, we're talking about 22 autosomes, which code for everything, and then you have one sex chromosome. Okay. The egg is 22 chromosomes plus an X chromosome, and the sperm is 22 plus X or 22 plus Y, and then these merge together. Okay. So here's like a picture if you want a picture representation of how this looks like. So a chromosome, remember it's like an X and it's bonded in the center called a centromere. And this little X, the reason why it's an X is they call these sister chromatids. So one of these is like a chromosome, and then the second part is like a duplication of that. And you may notice there's a blue and a red. In this case, the key says the red is the maternal set of chromosomes, so this one's like the moms, and these are like the dads. They both are sort of related to each other because they both sort of kind of code for the same thing. So blue and red, one from mom, one from dad. So here's this might be easier for you to see a picture. Another definition you need to know is called a karyotype. A karyotype is a picture of an organism's complete set of chromosomes. So it's literally, which is right here, a karyotype is literally a picture of just what are your chromosomes, and you could actually see your chromosomes if you really wanted to. How they arrange it, how they make a karyotype, they take all your chromosomes, which is here you see on this lady's TV, and they just literally just sort them. All the largest ones, the two largest ones go here, then the next largest ones go here, and they just sort them. So they pair the homologous chromosomes. So here's that X if I want a more detailed, zoomed-in version of it. Here's my X. Remember, my center is a centromere. These are sister chromosomes, so they're the exact same thing, but a duplication of one of the chromosomes. And this is, you get this over in metaphase. So here's a cool, quick example of a karyotype. So again, what I saw on that lady's TV, here are your chromosomes. Okay, just unsorted, just like that. And their whole job is to take them and sort them like this. Now the question is this, looking at this karyotype, is this person, it's cut off here on the bottom, but you'll see on your notes, is this person male or female? So remember, there's 22 autosomes, so if you could see there's 1, 2, 3, 4, 5, again, sorted by size. How you can tell this is a male or female is you look at the last one, not the 22 autosomes, but the last one, and you may notice that it has 2 X chromosomes. So since this person has 2 X chromosomes, this is a female karyotype. On the other hand, this next one, you may notice, same thing, 22 autosomes, but this one has an X chromosome, you notice it's long, and a short Y chromosome. This tells me that this is a male, because a male has an X and a Y. So the reason why they use karyotypes, or why we care about karyotypes, is, I don't know if any of you are interested, but when you have offspring, you can actually determine if there's going to be any genetic abnormalities before they're even born. So these are what a normal karyotype or normal chromosomes looks like. If it doesn't look like this, or you have an extra 6, or you're missing like a 16, then you know there's going to be a genetic abnormality before the baby's even born. So a life cycle, typical life cycle of an organism. So you usually have fertilization, which I know you all know, and meiosis, and they alternate. This is in sexual life cycles. So those that have kids, male and female, it's just alternation between fertilization and then meiosis. Fertilization, meiosis. So the life cycle, fertilization, meiosis, fertilization, meiosis. Meiosis, what they do, they call this reduction cell division. So mitosis, the one that we studied already, is duplication. You get what you start with, you just create a double of. Meiosis, what you start with, at the end of it, you get half. Okay. Fertilization, you combine the gametes. So for males, their sperm. For females, an egg. And once these combine, you get something called a zygote, which you've heard of before. The zygote, if you've seen like a baby, I don't know if you guys have ever been to the museum or you've seen it, a zygote's like one giant cell, and then it duplicates, and you get 2, and that duplicates, so it's constantly dividing by mitosis. That's how a baby is made. So here are the different sexual life cycles. We're going to go through 3 of them. Here's it for animals, here's it for plants, and here's it for fungi and protists. Okay, they kind of look the same. They're still meiosis and fertilization, but they're completely different, and we're going to go for these individually very quickly. The human life cycle is the easiest one for you because you know what a human life cycle looks like. So don't mind the naked pictures of a male and female, but in the testes of a male and the ovary of a female, they create a sperm and an egg. These are haploid gametes. Remember, these only have half the chromosomes. Okay, so normally we have double. Normally we have. Remember, we told you we have 23 pairs. These ones only have 23 total. When they combine, I get my 46 total chromosomes. And then it doubles and doubles and doubles, so you get here, and you start all over again. That's the human life cycle. So it's the same thing for animals. Okay, you start off with think of this as us, or a diploid multicellular organism. They divide meiosis, you get a half of your chromosomes and another half from somebody else. They combine, they fertilize, and then you're back where you started from. But it's a little bit different for plants and protists and algae. There's the term that you need to know is called the alternation of generation. So they actually alternate life between a 2N and an N. Okay. For plants, they call it a sporophyte if you have a 2N, or if you have like your full set of chromosomes, they call you a sporophyte. These sporophytes, they make haploid spores, kind of like how we make sperm and eggs, by meiosis. The spore then becomes a gametophyte by mitosis. So here's that same picture of a diploid multicellular organism, has meiosis, and now we have spores, which is like how we have sperm and egg. But the thing is that the difference is that they actually can exist as an organism in that spore form. They call it a haploid organism or a gametophyte. Okay, so our sperm and eggs, they're not like organisms. You don't just see like a random sperm or egg like walking around, or it's like, hey, it's normal to have a sperm and an egg, like a half chromosome, just live. But plants do that. Plants have a double diploid. They release spores, and then sometimes those spores become their own organism that just happen to have half of their chromosomes. And then these haploid organisms can combine with other haploid organisms. They fertilize and they get back to the diploid. So again, completely different than humans. Probably really confusing, but long story short, they just alternate lives, alternation of generations, between a diploid, or double chromosomes, and a half. So here's a picture. We'll skip over it real quick. Fungi, protists, and algae, a little bit different than plants. Same thing as us, but the funny thing is it's completely reverse. They don't even have a life as a double. So we exist, humans only live life as a double chromosome. Fungi, protists, and algae, they only live life as a haploid. So they have a haploid, this is where they start. They combine, they fertilize, and they have a zygote, and those zygotes release some sort of spores again, or half chromosome gametes, and then they live their life once again. They do not have a life or an organism where they have double the chromosomes. It just doesn't exist. Just like we don't have an organism stage where we only have half our chromosomes. So those are the varieties of sexual life cycles. So I need to go through this real quick. We're already running up against 12.5 minutes. Meiosis, again, is reduction division. Whatever you start with, you end up with half. Okay, the result is four daughter cells, each with half as many chromosomes as the parent cell. So here's like a basic overview. So you remember interphase. This is what a chromosome looks like. Remember, you just saw a karyotype. It's just like one little line. This one is from I forgot which one is. I forgot the key, but one's from mom and one's from dad. They duplicate in interphase, right? So this is normal. Here's it duplicated. In meiosis, what happens is that these homologous chromosomes, they separate. So this blue one goes to its own cell, this red one goes to its own cell, and then these X's are split in half. So now, what I once had, I have I have four cells, four daughter cells, that only have half the chromosomes. Okay, so going through this super quick. Meiosis 1 is exactly like mitosis. There's some very key differences. Okay, interphase you already know. Prophase, and this in meiosis, they have something called synapsis. Synapsis means each of the maternal and paternal chromosomes, they all line up together down the middle. Okay, here they are. This is just prophase, so they just pair up. It's not down the middle yet. A tetrad is four sister chromatids. So this right here is a tetrad. So you notice that when they come together, that's called a tetrad. And then they cross over. So you're saying, what is crossover? So the mom and the dad, so say for example, I'm being oversimplistic here, let's just say like this chromosome is all for hair color, and this one is all for hair color. Crossing over means like this, the dad part will actually merge and like cut and add to this part, and the red will actually cut and split onto the blue part, and I'll show you what that looks like. That's called crossing over. The chromosomes will actually exchange information. Metaphase is the same as metaphase and mitosis, meta meaning middle, they line up down the middle. And anaphase, they separate. These ones, they're still attached by the centromere. So they don't actually break apart like in mitosis. Just this one goes to this side and this one goes to this side. And then telophase and cytokinesis, you remember, they just separate and then it cleaves and it breaks into two cells. So here's a picture of it in prophase. Here's it in metaphase. It's right down the middle. Here's what I was trying to tell you about the crossing over part. So when they're in the middle, right here, in metaphase, they cross over. So see how this is completely blue and this is completely red? When they cross over, they exchange information. So over here, this blue now has kind of like a red piece, and this one now has a blue piece, and the crossing over is completely random. So this one actually has 2 blue pieces, this one has 2 reds, etc. And then telophase, it breaks apart. Okay. Meiosis 2 is something completely different. Prophase 2, there's no interphase. There's no crossing over, and the spindle forms. Metaphase, middle, they line up. Sister chromatids separate. Okay, so the centromeres actually bust apart, and then you get 4 haploid cells, where the nuclei reappear, and each daughter cell is genetically unique. Now, if you want to see how a picture it looks like, so this is what at the end of meiosis 1. Again, we have metaphase, where they line down the middle, meta meaning middle. Anaphase, these X's, those centromeres burst apart, so you may notice these X's are gone, and then they separate, and now you have 4 complete different cells that have half the chromosomes as usual. So here are events in meiosis 1 that are not like mitosis. Okay? In meiosis 1, in prophase, they have synapsis and crossing over. You do not get that in mitosis. Okay? So prophase 1, the crossing over piece is not in mitosis. So we're talking about what's so different. In metaphase, remember the meta meaning middle, they line up down the middle. In meiosis 1, they line up down the middle by homologous chromosomes. Okay? So in mitosis, these are just one X. They're just X's down the middle. Here you see tetrads, XX, XX, and an XX. And the last thing that's in meiosis 1 that's not like mitosis is that in anaphase 1, the homologous pairs separate. So see how they separate? They do not burst apart, and they're still attached. Meiosis 2 is exactly like mitosis. Like the steps are exactly the same. But in meiosis 1, these are differences that are not like mitosis. So lastly, sorry, I know we're running long. Sources of genetic variation. We're wondering like why are brothers and sisters? How can they be completely different? How are there genetic variations between the same parents? How come the offspring look different? There are different reasons. One's called crossing over, which we talked about. So at the chiasmata, what happens is that they cross over some of their information. So if you take a look at this picture, remember I told you the blue and the reds, they sort of cross information. They could cross a lot of information, they could cross very little information, but this crossing over guarantees, see, look at these daughter cells, how all the lines, this is all blue, this is blue and red, this is red and some blue, this is all red, basically guarantees that all the kids, like all the sperm that you have or all the eggs that you have will be completely different, okay, crossing over. Another source of variation is independent assortment of chromosomes. So what this means is that when you go down the middle in metaphase and you split apart, how those blue and reds are lined up completely can change how the kids look like. Look at these 2 possibilities. Possibility 1, see how the blues and the reds are neatly on their own side? All the blues go here, all the reds go here, and then when you get those 4 daughter cells, I have all blues, all reds, and all reds. So that's one random possibility. Same parents, one possibility could be like, oh, like when they line up, the red was actually here and the blue's here. How does that change everything? Well, now in metaphase 2, I have a blue and a red and a blue, and all my daughter cells will have 1 blue and 1 red. So even though they're the exact same parents, you can have completely different kids, and that's called independent assortment of chromosomes. Okay. And the last one is random fertilization. So think of all those daughter cells we've been talking about. Any different daughter cell can combine with any other daughter cell, and that's how you have like basically complete variation between all kids. So any sperm and any egg. I mean, there's 8 million versions of sperm, 8 million versions of eggs, which means there's 64 trillion combinations. Even though the exact same parents, you have 64 trillion different ways the baby can look like. So just to wrap it up, the difference between mitosis and meiosis you should know. We'll close on this slide. I know there's more, but long story short, you should know the difference between mitosis and meiosis. Okay. Today in class, you're just finishing up those notes. I know some of you that emailed me and messaged me said that you need more time for those packets. I know you had a block day, so I just want to give you that. But if you need more time, tomorrow listen to another lecture, a completely new topic. But let's just say by the end of class Friday, all those packets should be completed. If not, or if you're done already, you watch this video, that's all I have for you. And then that's it. So take care, and I'll see you for tomorrow's video. So take care.